is Pierre Gönczy. I'm a professor at the Swiss Federal Institute of Technology in Lausanne. And actually, Lausanne is a place I moved to after having spent 4 years at the EMBL. So I was here from 1996 to 2000. And to tell you the truth, I knew very little about the EMBL before coming here. I basically did something that people would probably not do today, which is sit for 2 months in the library and read many things to decide where I would like to do my postdoc. Came up with a list of 40 labs, which then my advisor told me it was probably a little bit too many. And then I went down to actually 10, which did not include the lab of Tony Hyman, because he was working on questions that were different than the ones that were of interest to me at that time. But my advisor knew him as because they were postdocs together at UCSF, and he said, well, since you're interviewing in Europe anyways as part of your world tour, why don't you also visit Tony, which I did. And that's how I ended up at the EMBL.
So what attracted you of Tony, since he was not
you know, that's a very difficult question, and I'm not sure that should be on camera. But I don't know. I do think that literally the day that I spent here was very exciting. I could feel. I think that I'm sort of trying to recollect this because I've never reflected on this question really. I know why I did not go to many other places, and I could tell you 10 interesting stories about that. But that should be off record. But I think I did find the vibe extremely exciting. And also what I was proposing to do in Tony's lab is something he wasn't doing at that time, and I foresaw that I could have a good time here, sort of developing something that I had been thinking about, which is sort of bring cell biology into a developmental system where there was genetics.
So maybe the question for you, since you were taking on a new system, I mean, how did you find the EMBL, and how do you think the EMBL enabled the research that then you ended up doing?
Yeah, I have to say that not in a direct manner, because actually there was no single C. elegans at the EMBL when I arrived, and there wasn't one for several months until I got the first ones from the lab of Jim Priest in Seattle. I set up also the the plate pouring facility at the EMBL. But I guess maybe it's more like the way EMBL made that possible is that it is an environment where you know that everything is possible. And so it's more like, you're in a playground and you can basically reach out for any toy and you can make your own toys. And that I think is important.
And I mean, what I found amazing is to discover that there was no C. elegans before. And if I recollect from your time here, you went from that to actually screening essentially genome-wide the C. elegans for, mutants that would affect the early development, which is, it was then and it still is quite a monumental.
Well, it was, but it's the fortunate conjunction of various, we heard about serendipity this morning at the symposium, and I think I was just lucky to be at the right place at the right time for a number of reasons. And some of them interesting, some of them just really coincidental. So I think, first of all, I had developed an assay to recognize, I should say that I was interested in understanding how fundamental cell biological processes are deployed during early development. Okay, this was my general question. And there were several sub-questions in that general question. But there are basically 3 things that enabled me to develop this approach, which is, again, when you ask a kid from the kindergarten to think about this, they would come up with the same ideas. So I don't think the idea is brilliant, it's just obvious. But I just had the luck and the good fortune to be at the right time at the right place. So number one, I had, through screening mutants of early C. elegans development, literally going through the garbage can of other people who are not interested in these mutants, I was able to show that just by doing time-lapse microscopy, I could reveal very specific phenotypic classes in these early embryos that weren't doing well and eventually died. Okay, so I had this assay which was very simple, but very powerful in telling me what went wrong with every single strain. Second, at that time, the C. elegans genome was the first metazoan genome being sequenced. Okay. And so this was an amazing window on life, which had, been seen before in eukaryotes in Cerevisiae, but this was the first metazoan organism whose genome was sequenced. So we suddenly had a compendium of genes to target, and 3, RNAi came along. The initial report of RNAi in C. elegans is 1996, a Cell paper by Su Guo, PhD student with Ken Kenfuse, where they say basically, and it's a super interesting story, but they basically say we have this bizarre phenomenon where we can phenocopy the PAR1 mutant phenotype by injecting both sense and antisense RNA. Makes no sense, right? And then Craig Mello and Andrew Fire published in 1998 this paper where they excised the double-stranded contaminant from the reaction and realized that that was the active species. Anyway, so I was at the EMBL at that time, so having this assay, having the genome that came out, and having RNAi made it totally obvious to me that we should just simply, quote unquote, inactivate every predicted predicted open reading frame from C. elegans and use this assay to determine what function could be ascribed to these genes in the early C. elegans embryo. So that was really simple. There's a little EMBL Max Planck story here as well, because making, at the time we were making double-stranded RNAs in vitro, okay, and injecting them into the animal. And that meant that this cost a lot of money. So in fact, the fact that Tony and the others had been named Max Planck directors but were still at EMBL made it possible for me to access funds that I would not have been able to access at EMBL at that time. And I remember of this meeting where I had to present this project to the 5 Max Planck directors in a little room across from the ISG in Boxberg here. And I just said, I need 200,000 Deutschmarks at that time just to do one chromosome. They said, no problem. So that was really good.
But you pointed out that you probably were in the right place at the right time. But nevertheless, also the way the screen needed microscopy, needed parallel processing and everything made for a very interesting and challenging scale up of biology, and I think
super interesting.
Yeah. So I want to hear a bit about that, and maybe since I remember that this became even a startup, right? And it was the first time maybe that EMBL was experimenting with startups. So maybe a little bit of your thoughts of why it came up that way, what then achieved, what was the importance. Let's talk about this thing.
Yeah. So I think the idea of scale up, which of course now would be trivial for any high throughput screen, at that time was not very much the type of thing you would think about. So when we wanted to do basically one chromosome as a proof of principle. And we were racing against some deadlines due both to conferences, competition, and what have you. And indeed, for a month, we had basically a 24/7 schedule with 3 8-hour shifts. And we were, again, thanks to the Max Planck funds, able to buy 6 microscopes and basically did everything highly parallelized. interestingly also, I think, and this came later through the work of others, this also then yielded a need for analyzing the data in a manner that, could make sense in terms of clustering phenotypes, in terms of deriving relationships, which again nowadays would be trivial for anyone doing, let's say, CRISPR-based screen or whatever. But at that time, we were sort of inventing that language in a way.
Yeah, and what about the startup idea?
Yeah, the startup idea. Okay, so the startup idea has very little to do with me, but I'm happy to talk about it anyway. So the startup idea is basically Kai Simmons, who told Tony that this should be a startup. Maybe because he was worried of how much the whole genome would cost to the Max Planck. I don't know, that's just an inference. And basically, he came to me and asked me whether I would be interested in, starting a company. And I told him no. And so then he asked Chris Echeverry, who was also a postdoc in the lab, who contributed importantly to the screen as well, who was interested. So then Chris, Tony, and I co-founded this company, which went bust now, but which I'm very happy basically gave jobs to 30 people for 15 years. And I think that's quite fantastic, actually.
One could also argue, as you were saying, that this was maybe one instrument to do that kind of research that otherwise would have been maybe too expensive to be done, or to focus on that type of systems biology, early systems biology.
Yeah, I mean, I think the screen would have been done no matter what, but maybe with a different pace. The company's business model was both work as a service company. Basically, at that time, not everyone, including pharma, could do RNAi-based screens, including in human cells and everything. So that was part of the business model. I think initially they were also hoping that some of the genes that we patented could be utilized as anti-parasitic, targets for anti-parasitic compounds, for instance, genes that do not have obvious homologs outside of nematodes and that are essential for C. elegans life, were sort of interesting targets potentially.
Yeah, and so, this is more generally related to your research, but you and I, we both work on model systems and animal models, and so now animal models are challenged because people are thinking about the ethical consequences and a potential other system that could replace animal models. So I was wondering whether you can reflect a little bit, being working with C. elegans, on the role of animal models, and what is your view? Should we just move to other systems eventually, or do you still think there is value in using animals?
The answer to that question is extremely obvious, but I just want to, and the answer is, of course, we should work with whatever we want, and discoveries will come from corners that we don't expect. And the history of science has said that, and there's no reason that I foresee that changing at all. There's animals and animals. For instance, in Switzerland, flies and worms are not considered animals from the point of view of the law or ethics or whatever. So I think the considerations are a little bit different from that, and maybe sacrificing monkeys or mice or whatever. And I respect those differences. So I would rather, maybe if I can rephrase your question, I would say, is there still usefulness to work with model organisms? And the answer is obviously yes, for the reason that I just said. History teaches you that unexpected discoveries come from unusual organisms, and let's say CRISPR-Cas9, for instance, is an obvious recent example. But So that's the main thing that I think we should remember.
I mean, surprisingly enough, I mean, also in Italy, of course, fruit flies and worms are not ethically regulated or animal welfare regulated. But I heard a lot of calls for Drosophila because it is expressing very complicated behaviors. Certainly, there's our aspect of reactivity and pain.
I completely agree with that.
Yeah.
And in fact, in Switzerland, we have this interesting habit of voting on sometimes very absurd things every now and then. And about every 5 to 7 years, there's something from the animal rights people to sort of prevent, the use of animal systems to various degrees. And the last campaign was maybe 3 or 4 years ago. And our communications service has decided to be, rightfully so, to be extremely open about what we're doing and sort of publicize how animals are used and everything. And they chose one day to put, our work on C. elegans on their webpage. Mm-hmm. And I had to explain to them that technically, in fact, indeed, C. elegans is not considered an animal that actually sort of reacts or senses or feels. But of course, this is a fallacy because, not only flies sense, but, many touch receptor, pain receptors were discovered in C. elegans.
Exactly. So maybe, the answer is just keep on telling these stories and the public will say, well, I have concerns, but certainly there's a value in that. So that could be.
Yeah. The other question also maybe to that concerned public is, when you're bitten by a mosquito on your arm, do you actually try to squish that mosquito or do you try to save it and let it fly away?
So coming back to research, then of course you left the EMBL and you moved to your next position. So I wanted to know how the EMBL helped you in the following part of your career. I mean, did you apply some of the lessons learned at the EMBL? Did you try to replicate some of the EMBL environment?
I was wondering about that. No, I don't think in a conscious manner, but many of the things I now realize, having thought about it no longer than this morning, prompted by yesterday's email, that perhaps more than I thought. So for instance, I started a PhD program at my institution together with another EMBL alumnus, as it turns out. And in our school, we have, for instance, a philosophy of building joint platforms, which again is not unique to EMBL, but it's the first time I had seen it actually operate so well here, where, and so, perhaps there is that spirit that actually percolated. But it's not that I've gone to my current position and say, okay, what do I need to build like EMBL? I think it's more like, I think I, as a person, fitted extremely well to the environment here. Mm-hmm. I think maybe helped to shape it in my little minor way as well. And some of that I've taken with me, but it hasn't been conscious.
Okay.
So
and how did your research evolve? I mean, what are you passionate about today?
Yeah, right now my research has evolved a lot towards centrioles. So we've been extremely obsessed with centrioles, and actually this comes back to research that I've done during the RNAi-based screen here. Because one of the 2,000 and some genes that were located on chromosome 3 is this gene that we eventually termed SAS4 for spindle assembly defective 4. I had identified SAS1, SAS2, SAS3 by mutational analysis, so this became SAS4. And the phenotype was extremely interesting and suggested very strongly that this gene, when inactivated, prevented the formation of centrioles. And I knew about centrioles from my reading, from my background, and also from a mutant in C. elegans that had a very similar phenotype, work from Kevin O'Connell at NIH now. And this seemed extremely interesting because nothing was really known except for that mutant in C. elegans, which had just been published after I started my lab, but which I knew about from conferences. But really, essentially, nothing was known in any system about how centrioles are made. And centrioles are fascinating, they're beautiful. People who are proponents of intelligent design have written books, literally, I've read parts of them, that use the shape of the centriole as proof of the existence of God, because there's no way that evolution could have come up with something that beautiful.
And then you work to disprove that, of course.
And
certainly, I have to say that they are fascinating and they are a mystery, and whether they are remnants.
No longer a mystery. So in C. elegans, I mean, we through this screen, through also mutant screen that we did, we as well as others we're lucky to identify basically 6 core components that are essential to make centrioles in C. elegans. And it turns out that they are conserved across eukaryotic evolution. And that means they've been on this planet for over 2 billion years. And if you get rid of them in clammy, in human, in C. elegans, same phenotype, you don't make centrioles. And that's essentially the thing they do. And we have then, of course, gone from having identified these components to trying to understand how they function. And so this has become an obsession of my lab, trying to understand how centrioles are formed, what regulates their loss. We've published a paper last year where we discovered that C. elegans actually undergoes programmed centriole elimination. 88% of cells during embryogenesis, always the same ones, lose centrioles at a given time. We're interested in understanding how centrioles are eliminated during oogenesis. They must be eliminated during oogenesis, otherwise the embryo receives too many centrioles and fucks up basically the first mitotic division.
Cytoskeleton organization.
Yeah, and it's sort of a converse of mitochondrial inheritance if you think about it. So our kids, we got our first centrioles from our dad, not from our mom. And we've done this fascinating experiment in C. elegans, very simple-minded, where we've asked how long do the paternally contributed centrioles persist in the embryo? And they persist for a very, 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 very long time, unless they end up by chance in the 88% of cells where centrioles are targeted for elimination.
So I remember from the very past, but I might have dreamt about that there was somebody saying that this was the remnant or potentially of again another symbiont or a bacteria. Is that story still true? Nobody knows.
It's really interesting that you bring up this point. My lab has been very interested recently on trying to understand the origin of centrioles. When you read about centrioles, people say they appeared in LECA. Things rarely appear. I mean, Pasteur sort of told us that this is very unlikely in general. And you don't find a trace of these proteins in the currently sequenced archaea, bacteria, or viruses. Okay. So we're trying to,
So there's still a lot to be learned.
Absolutely.
Yeah. No, that's great.
As usual.
I think what has been fascinating is that you could see the structure by electron microscopy, but somehow is one of these things that you cannot really define as an organelle because it doesn't have a membrane. Certainly there is a lot of architecture about that. So somehow the funny reflection in thinking today about Tony Hyman's lecture is one of these subcellular structure that is difficult to define, and it remind me again, although they are clearly probably not liquid particles, it remind me again of one of these cellular structure that because you couldn't see them completely remain kind of elusive. You could see only the internal structure.
Totally. I mean, it's in 1958 that the first basically paper about the structure of centrioles was published, because until then, I mean, they're so tiny, right? half a micron long if all goes well. In C. elegans, they're cube-like, 120 nanometers high and 120 nanometers across. Who would pay any attention to them? And yet they're so fundamental.
So because we are having these festivities for the 50 years of EMBL, I would like to ask you what EMBL means to you.
Home.
I can relate to that. I always call it the mothership.
Yes. It's not my only home, but it's definitely one of the homes. Yeah, and I think part of that is, of course, you have connections, with the place where you've spent some very important formative years. I have very strong connections also with my alma mater in New York, but I think here the fact that we have conferences, the fact that there are so many activities that bring us back here from time to time, obviously participates in strengthening this bond and keeping it over the long haul.